Hello, welcome to PalaeoPERCS. So I think we're just gonna let the last couple of folks filter in, but welcome to PalaeoPERCS. Good morning, good afternoon, good evening, depending on where you are in space and time. My name is Elizabeth Seibert. I'm one of the PalaeoPERCS committee members, and today it is my great pleasure to introduce Flavia Boscolo Galazzo, who will be giving a talk on her recent science paper, Temperature Controls, Carbon Cycling, and Biological Evolution in the Ocean Twilight Zone. Flavia is currently at the Bergen University in Norway. And so just a quick welcome to those of you who have not been in a PalaeoPERCS seminar before. We'll just do a quick set of welcome and announcements. Then Flavia will give about a 30-minute talk. We'll have a moderated question and answer session, and then Flavia has agreed to stick around after the talk for about 30 minutes to have a tea time, and we really encourage you to stick around and have a conversation with her. And we'll just mention that you should send any questions that you have or any technical issues that you've got to the questions at PalaeoPERCS host, who I believe is Raymat today, and so she'll help out with that. Just a couple bits of housekeeping here. We want to remind everyone that PalaeoPERCS values the participation of all folks interested in paleosciences, and so we'd like to ask you to please remember to abide by our code of conduct during today's seminar. And if you have somehow managed to pop into today's seminar without reading and checking off the code of conduct, please navigate to our website and just take a quick peek at that. We also like to ask you to please mute yourself for the duration of the talk, and remind everyone that questions are very strongly encouraged, and that we would like you to ask questions by chatting them directly to the PalaeoPERCS questions host, and we will then read them out during the question and answer. If you could also mention when you send your questions in, whether you'd like your question to be anonymous or read by name, we can make sure that happens as well. A couple of other little bits of housekeeping. We're currently using closed captions through Zoom. And so if you want to hide those captions, at the bottom of your Zoom menu, there's a CC button. You can click that and choose Hide Subtitle. And then finally, if you wouldn't mind filling out today's SurveyMonkey demographics survey that has been dropped in the chat. And it's totally anonymous, totally optional, just a chance to let us know who's coming to these seminars so we can do a better job of reaching the community and get what we need in here. I'd also like to mention that we are always accepting nominations for excellent early career scientists to give talks. And so please feel free to nominate or self-nominate an excellent early career researcher at our website. All right, so that was a lot. So without further ado, I'd like to bring us back to Flavia. So Flavia did her bachelor's, master's, and PhD at Padova University in Italy before heading off to the UK to do a postdoc at Cardiff University. And she's currently a postdoc at Bergen in Norway, but is actually going to be moving to Germany in 2022 as a newly minted Marie Curie Fellow. So I'm really excited to hear her talk today, and without further ado, I will invite Flavia to take over the screen share, and we will hear about the twilight zone and carbon cycling and evolution.
Thank you, Elizabeth. Yeah, so thank you very much for the nice introduction, and thank you very much for having me here today. So yes, today I'm going basically to present our science paper. As you may know, the results of this study have been recently published in Science. So yeah, this was a NERC-funded project, PI Paul Pearson. The work I'm presenting today was carried out by myself and by Catherine Crichton, who developed the modeling component of the study and who's now based at Exeter University. So global cooling controls carbon cycling and biological evolution in the ocean twilight zone. So I wanted to start by sharing with you this triptych of painting, as I think they very efficiently convey the paleontological angle we adopted in studying the ocean twilight zone during this investigation. They represent 3 snapshots of time, 15 million years ago, 4.5, and the modern ocean, and they show planktonic foraminifera living a different depth habitat through time. And this basically summarizes the finding of our study and also what we think has been the evolution of the ocean twilight zone over this time interval. So because planktonic foraminifera are so central to this talk, I decided to start by giving a quick overview of what we know about modern planktonic foraminifera ecology. So planktonic foraminifera are protists. They produce a calcareous shell which easily fossilize, and that's why they are so widely used for paleontological reconstructions. Modern planktonic foraminifera can be roughly divided in 2 main ecological groups. Surface dweller species and deep dweller species. Surface dweller species are largely spinous species. They have obligate symbiosis with algae, and that's why they preferentially live in surface waters. They are also, at least many of them, carnivorous, and they prey on zooplankton such copepods. Subsurface and deep dwelling species, they don't have symbiosis with algae. They are largely omnivores, and they rely on the settling of organic matter from the surface as primary food source or as the food source of their prey. So this is of course quite schematic, but I think it's important to bear in mind throughout this talk. So the aim of this project was testing 2 main research hypotheses. The first research hypothesis we wanted to test is whether secular changes in ocean temperature can modulate the efficiency of the biological pump in transferring organic carbon from the surface to the deep ocean on long geological timescales via metabolism temperature dependency. So, but what is metabolism temperature dependency, and how does it fit into the picture? So metabolism temperature dependency is basically a physiological response whereby for each 10 degrees of environmental temperature increase, organism metabolism nearly double. And you can see this here for one and multiple species of fishes. The higher the temperature, the higher the organism consumption rates. And this applies for many different types of organisms and almost any biochemical reaction, including photosynthesis and respiration. So if we transfer this to the ocean biological carbon pump, this may imply that during overall colder climate, more organic matter can make it down the water column into the deep ocean because of very low bacterial metabolic rates at the surface. Vice versa, during overall warmer climate, very little organic matter could make it down the water column to the deep ocean because of very fast bacterial metabolic rates at the surface, very efficiently remineralizing organic matter already at the surface. So if this of course can imply can have a wide implication in terms of CO2 partitioning between the surface and the deep ocean, but also for all those life forms which populate the ocean below the euphotic zone and therefore rely on organic matter sent from above for their survival. And this links us to the second research hypothesis we wanted to test. So because we know that the planet has been largely cooling over the past few million years, what we wanted to test is whether a more efficient biological pump in a colder ocean may have triggered the evolution of modern deep water niches. So to test our research hypothesis, we used a combined data modeling approach. We obtained data from planktonic foraminifera, as I'll show you in a minute, and data simulation from an Earth system model of intermediate complexity called Gini. So we focused our investigation on a time interval covering the last 50 million years because we know that over this time there was an average cooling of the global ocean between 6 to 8 degrees, and therefore this time interval represented an ideal case study to test our research hypothesis. In particular, we focused on 7 target ages between 15 and 0 million years ago, which you can roughly see here. In terms of geographical coverage, we worked on a range of globally and latitudinally distributed sites, which you can see here, both in terms of data collection and model simulations. However, these sites were primarily selected based on planktonic foraminiferal test preservation, as we were keen to work on planktonic foraminiferal specimen with very good to excellent test preservation, because this is very important to reconstruct water column chemistry. So data-wise, we obtain assemblage count, so species-specific assemblage count for each sample, so for every site and for each target age. We try to count as many different species as possible. And also we measure oxygen and carbon stable isotope on as many species as possible. I'm showing here just a couple of examples of our multi-species oxygen and carbon stable isotope data for a couple of our investigated sites. And this is what I've been working on. Meantime, Katrin developed 7 different configurations for Gini, 1 for each of our 7 target age of interest, and each of these with different and time-specific boundary conditions. And here with boundary conditions, I mean, for instance, atmospheric CO2, continental configuration, ocean circulation, and so on. But the very important thing to bear in mind here is that she developed a new temperature-dependent configuration for the biological pump in Gini, where nutrient uptake rates and organic carbon remineralization rates are modulated by temperature, are temperature-dependent in the model. Vice versa, in the standard configuration of Gini, nutrient uptake rates and organic carbon remineralization rates are constant and do not change with temperature. So the results of this different configuration you can easily see on these 2 projection maps for the flux of organic matter at 1,000 meters depth in the modern ocean. In the temperature-dependent version of Gini, this parameter is much higher in cold subpolar regions as opposed to the tropics. Vice versa, this same parameter is constant across the ocean in the standard version of Gini. So to test our research hypothesis, we performed 2 different tests. For the first test, we look at temperature effects on organic carbon remineralization rates. And for the second test, we look at changes in biodiversity and biomass at depth over the last 50 million years. For the first test, we used our multi-species delta C13 data from planktonic foraminifera to reconstruct delta C13 DIC dissolved in organic carbon gradients for the upper part of the water column. This is because at these steps, this gradient is largely controlled by the balance between CO2 uptake from photosynthesis and CO2 released by organic carbon remineralization. We then compared our reconstructed delta C13 gradients with modeled gradients obtained from both the temperature-dependent version of Gini and the standard version of Gini. For the second test, we used our multi-species delta 18O data from planktonic foraminifera to reconstruct species-specific paleodepth habitat in combination with Gini water column temperature and depth profiles. We also used our assemblage counts in combination with the species-specific paleodepth habitat reconstruction to reconstruct planktonic foraminifera biomass depth distribution through time. So let's start looking at some data. So to start with, I'm showing an example of how we calculated the delta C13 gradients the delta C13 gradients. Yes, so this is for one site at specific target age. This is depth and this is delta C13. The black squares here represent delta C13 data from different planktonic foraminifera species. The red triangles represent delta C13 output data from the temperature-dependent version of Gini. And the blue circles represent the delta C13 output data from the standard version of Gini. As you can see, we consider here only the upper part of the water column. We performed linear regression for each of these different data pools and did so for each site and for each target age, and then obtained global mean values, which I'm showing here. So here the gradients is represented as a water column change in meter per 1 per mil change in delta C13 versus time. So here the red symbols represent the global mean values from the temperature-dependent version of Gini, and the blue symbols represent the global mean value from the standard version of Gini. And as you can see, these values are very different from each other, and they also show opposite trend through time. In particular, the temperature-dependent version of Gini show a slightly deepening trends through time, which means delta C30 ingredients change less with depth. And this is driven by a decrease in organic carbon remineralization rates, a temperature-dependent decrease in organic carbon remineralization rates in the model. So if we now compare this with reconstructed delta C30 ingredients from planktonic foraminifera, we see that they are strikingly similar to the temperature-dependent version of Gini global mean values. The global mean value from reconstructed from planktonic foraminifera also show the slightly deepening trends through time, which is consistent with decreasing organic carbon remineralization rates with cooling as projected by the temperature-dependent version of Gini. So let's now have a look to biomass and biodiversity distribution at depth. So here I'm showing paleo-depth habitat reconstruction at 3 different sites. This is a mid-latitude site in the Atlantic Ocean, a tropical site in the Indian Ocean, and a tropical site in the Pacific Ocean. Again, this is water column depth, and this is time. Here, the gray symbols represents planktonic foraminifera species living in the upper 200 meter of the water column, and the multi-shaped colored symbol represent planktonic foraminifera species living in the twilight zone. And as you can see, twilight zone species remarkably increase in number through time. And also these deep dwelling species increase their depth range through time. And this is very nicely exemplified by the depth habitat evolution of a couple of species, Globorotaloides hexagonus and Hirsutella scitula, which are here represented by these pink and green stars. So as they first appear in the record, these 2 species progressively move to greater depth through time, up to below 1,000 meter depth in the Holocene. So let's have a look to biomass as well. Here, biomass is represented how the percentage of the whole planktonic foraminifera community versus depth and versus time. And as you can see, there is very little living below 200 meters depth at 15 million years ago, but then the proportion of the planktonic foraminifera community living at twilight zone depth gradually increases through time, and we estimate a doubling of the whole planktonic foraminifera population living at twilight zone depth by the Holocene. This increase in the proportion of planktonic foraminifera living in the twilight zone actually coincide with an increase in organic carbon flux at mid-twilight zone depth as projected by our temperature-dependent model. These are the plots for each of our investigated sites, and as you can see, these parameters steadily increase through time. So basically, our combined data and modeling results indicate a decrease in organic carbon remineralization rates in the upper ocean, and in parallel, an increase in the efficiency of the biological pump with cooling. This in turn was associated with an expansion of biodiversity and biomass at twilight zone depths. So, but what do we know about life evolution in the ocean twilight zone over this time interval from literature? So this planktonic foraminifera phylogeny actually tell us that the last major radiation among planktonic foraminifera happened roughly over the last 7 million years. And if we specifically look at which group evolved at this time, we see that they were the Globoconellids, the Menardellis, Globorotalis, Hirsutellis, Truncorotalis, Puleniatinids. These are all deep water groups of planktonic foraminifera, meaning that modern deep-dwelling planktonic foraminifera species actually started their evolutionary history back to roughly 7 million years ago, when we first start seeing this increase in the efficiency of the biological pump and depth habitat expansion. So there are actually evidence that something similar may have happened also for bony fishes. This is a phylogeny based on molecular clock data, which shows how the majority of deep water fishes appear to have started their evolutionary history between 20 and 10 million years ago, which, considering the different methodology applied by the planktonic foraminifera in this phylogeny, the results are quite consistent. And by the way, this also includes the iconic angler fishes. Yeah, so also ctenophores or comb jelly, they appear to share a similar evolutionary history. These are other common inhabitants of the twilight zone of the ocean, and all the modern species appear to be closely related to each other, which indicate a recent diversification, despite the group itself has a very ancient split from other metazoans going back to the Cambrian. So, and eventually, if we look at what happens at organisms living at the seafloor, benthic foraminifera shows an interesting pattern as well. These are data which are replotted from a paper by Thomas Egode in 1996, which shows the global abundance of phytodetritus species. These are species, benthic foraminifera species, which blooms at the deposition of large algal aggregates at the seafloor, forming large population. And therefore, and as you can see here, they increase globally, starting about 60 million years ago, and therefore they also seem consistent with an increase in the efficiency of the biological pump in delivering organic matter at depth and eventually to the seafloor. So to conclude, our combined data and modeling results indicate decreasing organic carbon remineralization rates in the upper ocean in parallel with global cooling over the last 15 million years. This went in parallel with an increase in the efficiency of the biological pump, meaning more organic matter was able to reach the twilight zone of the ocean. We proposed that this improved habitability with cooling was critical to open up new ecological niches at depth and for the establishment of the modern twilight zone ecosystem. And thank you very much for your attention.